Welcome to the first Build a Cell seminar of the 2025, and thank you, Stefan, for agreeing to give the seminar, and you can get started.
Welcome, everyone, and today's lecture will be on programmed biomembrane fusion, and this is obviously a highly relevant topic in general, and I will take you on a journey on how to do that by non-natural means, and try to highlight the relevance of biomembrane fusion in all processes associated to it. But before I get going with the scientific part, a short introduction to who am I and where I'm actually located. You see this little European country over here. If you skip Greenland, we are actually a rather small country. We have 4 major universities, and University of Southern Denmark being one of them. We have a healthy 25,000 students, and we have just tripled in size since we got a new huge hospital added to our university. And now everything is under one roof, which is really fantastic for people doing medicinal chemistry and translational research. And that will become relevant when I talk about applications on natural lipid nanoparticles a little bit later in my lecture. But let's go straight to the topic of interest. Before I talk about details about our approach to program biomembrane fusion, there's no doubt that life as such depends on compartments and the control of compartments. And in nature, this is obviously done by lipid bilayers and sort of fusion and budding off of compartments. And this happens all the time and is tightly regulated. And among the proteins responsible for that is a special group of snare proteins and some other helper proteins, which typically regulate these processes. And I've stolen this slide from a Nobel laureate lecture on the action of snare proteins. And you can see this is also visualized in a type of zipper action, very much like a zipper on a jacket. Important already on this simple slide is that it's not just about membrane fusion. Membrane fusion is only relevant in the context of content mixing. So the transfer of content is the key thing. And there are a number of interesting processes in the context, and I will just highlight one which we have altogether spent a couple of years on. And this is the action of how sort of a COVID-19 virus actually enters your cell. There is obviously recognition by the spike proteins, but they actually open up and deliver the actual fusion machinery to fuse the virus with the host membrane and, of course, the release of content. And as you can see, these are rather big and very complex proteins which facilitate this process. And they are not very amenable to a sort of supramolecular approach. If you want to mimic this process, there are groups working with snare mimics, but it's not a very flexible and certainly not a programmable approach. And we will come to the point why the programmable part is so important. And before I get to some of the other subjects shown here, this is sort of an overview of biomembrane related research in my group. We have been very interested in the very basics of simulating some aspects of biomembrane fusion, of applying that to multi-step kind of fusion events, looking at the smallest of scales and how many of these fusion systems we use are actually involved in these processes. And at the end, some real-world applications on both synthetic and natural lipid particles. So let's start out with our take on snare-mediated fusion and how this may help in simulating a part of these processes. Why are we bothering with that? Obviously, there are a lot of interest in working with biomembrane fusion. We have a particular interest in our group in the context of therapeutic applications, loading of natural particles such as exosomes, cells, and the like. And I will come to that at a later point. But before we look closer into applications, we need to understand some of the basic sort of activities which occur during fusion. Biomembranes are typically rather stable, but mechanically very fluid, very flexible. But there are barriers to the process of fusion, and it's very regulated. And it's typically assumed that you, as a first step, need to bring membranes into proximity. This is typically done with membrane anchors, which can be very complex, like in the case of snare proteins. And then surfaces which are very close to each other, that means sort of below a nanometer, they disturb the lipid bilayers. There's some dehydrogenation, and this lowers the barrier to actually fuse and ultimately mix content, which is the key step. When I talk about fusion, I always mean full membrane fusion with content mixing. Obviously, we have not been the first one interested in the subject, and we have also not been the first ones to work with sort of snare mimetics, as we like to call them. And I've just highlighted some of the very important groups which have been active in the field, and I apologize beforehand if I have omitted any other major contribution. There have been the work from the group of Frederick Höck and Andreas Hermann, and they have worked with sort of this sort of membrane anchors as part of DNA constructs. Then there has been the group of Ulrich Dietrichsen. He has unfortunately passed away, and they have worked with peptide nucleic acids, which sort of hybridize like DNA, but have entirely peptide backbones and subunits of natural membrane anchors. Then there has been the work of Alexander Gross, which used coil-coil peptides. They fuse very nicely, but they are not programmable. And then we have the work of Steve Boxer from Stanford University, which collaborated with Eric Kuhl on lipidated oligonucleotides. As you can see, lipidated oligonucleotides on the left and the right have been very important contributions. And they are the most interesting ones for us as well, because they are programmable in the sense, in the intrinsic hybridization capabilities of DNA, RNA, and actually also PNA. So what are the key challenges when we entered the field? We have been working with lipidated oligonucleotides before, so the chemistry was something we had already some idea of, but there are a lot of other aspects of biophysical nature which we had not much experience with. From the systems reported and from the systems we sort of tested from other groups, all of them showed very low level of content mixing, especially for the DNA systems. What does that mean? That means that we either couldn't detect it or it was well below 5%. And that is sort of in the background. Especially for the PNA systems, we have seen a lot of leakage, and this little video illustrates what the problem is here. The peptides are also not very programmable. So they are interesting to do fusion, but they are not very flexible as soon as you are interested in multi-step fusion. And for some of the candidates, also relatively high concentrations are necessary. I will come to that point as well, what high concentrations actually mean in the context of fusion. So what you really want is that you fuse membranes, and ideally 100% of the content is mixed with the new compartment with very little to no leakage. This is sort of the general design. This is the zipper technique, which is sort of involves two complementary strands of oligonucleotides in the broadest terms. I will show you examples for both DNA, RNA, and PNA, and lipid membrane anchors. And that may seem very simple from the outset. You just take a DNA and a lipid membrane anchor. But what you do get if you just terminally modify oligonucleotide with any sort of lipid membrane anchor is a highly amphiphilic molecule, which typically will self-assemble, will show strong surface or surfactant properties, and will not very amenable to controlled fusion of biomembranes. And we have worked with a lot of chemistry in this context to find the right membrane anchors and the right design to have oligonucleotides, which can do what we want them to do, and at the same time don't show sort of surfactant properties or self-assembly. Here are some examples. I have sort of skipped the DNA and just shown sort of the chemistry behind. Typically, you need lipids of a certain length, and you need typically more than one, even if you only want to membrane anchor or surface anchor a relatively short stretch of DNA. A short stretch of DNA in my world is anything below 50, and for longer oligonucleotides, you need a lot more membrane anchoring power. We'll come to that as well. And we have had, of course, a start with the first and sort of second generation, and they have been derived from a different project. They are excellent membrane anchors, but as you can see from the structure, if some of you are a little bit chemistry minded, they are not exactly easily accessible. So these are 15-step synthesis, and yields are not very good. And overall, there's a lot of effort. Nowadays, we work sort of with a sort of a third generation type of building blocks with a reasonably short synthesis and a relatively good yield. And I'm not sure how many chemists are in the audience, but this is a very reasonable number of steps. They are all very high yielding. This is from an older publication, so yields are typically better for some of the systems we have. And the last two steps are required anyway because they introduce the functionalities which are needed that they are compatible with automated DNA or RNA synthesis. We shorten them as LINAs, which stands for lipidated nucleic acids. Actually, a shortcut we have used by now for almost 10 years in literature, and I think it's also picked up by colleagues in the field. And I've only shown a very, very small section because this is not a chemistry lecture. We have really done a lot of different structures, and you will be surprised how many structures look like perfect membrane anchors, but they can't actually do the trick when it comes to actually look at diffusion. So distances are very small. So this is shown in angstrom, and this is sort of a semi-scientific depiction of a handful of oligonucleotides. And that becomes very interesting when we look at the microscopy later on, that the couple of duplexes I show is actually a real number of DNA copies you need to actually do fusion. So we are anywhere between 5 to 10 oligonucleotides. That means effectively 5 DNA pairs or RNA pairs to do fusion. These are, of course, very low concentrations, and it's very impressive on a supramolecular level that this is even possible. Talking about possible, obviously, we can make a lot of claims. We had to show that we actually can do fusion, and we had to adapt a handful of assays from the literature to our needs. SRB is a particular dye which is self-quenching at higher concentrations, around 10 millimole, and becomes very fluorescent upon dilution. And this was an assay which was used in literature, and since at the time when we started, we were new in the field, we wanted to be as compatible with existing literature to be accepted. So we adapted this SRB assay, and upon fusion, there are various options, and I will come to that. First of all, you need to have a look at some of the states which are involved, sort of textbook states which are involved in the fusion process. You have two populations you want to fuse, and the only difference between the two liposome populations are that one is filled with self-quenching dye, so they have a very, very low fluorescent background, and a population which is actually only contains buffer in the inner void. In case you have only assembly, nothing will change because all concentrations are unchanged, and the only thing you get is an assembly of particles, which you eventually can follow by UV spectroscopy just by absorption. The next step is hemifusion, when only the outer bilayers fuse. Again, nothing changes, and the only difference is that this process is not quite as reversible as the assembly process. And the ultimate goal, which is not reversible, is the actual fusion. And only during fusion and content mixing, we actually do see dilution of the dye. The very rigorous among you will say, how can you see the difference between dye which is diluted by fusion and dye which simply leaks out of the fusion pore? And this is a very, very relevant, important question which we ask ourselves. And we adopted a second assay. And this is the first assay I've just shown. And here you will sort of see the global content transfer of the SRB dye. But you can't really distinguish between what has been diluted internally in the new compartment and what has been leaked out to the surrounding solution. And to investigate that, we always do both assays at the same time with two different populations. And in this case, we have two populations which have both the same SRB concentration. That means in case they do fuse, nothing changes concentration-wise, and the fluorescence pattern is exactly the same. Only dye which may leak out in the process will be seen in this assay. And by subtracting the fusion, the total apparent fusion and the leakage from each other, you have sort of an effective content transfer. And this is what we use throughout the lecture. Okay, let's have a look to the design and what we looked at and tested and how we actually ended up having really efficient fusion at the end. And like with most project, our starts were very frustrating to say the least. Again, I have only selected a minor bunch of oligonucleotides to show you. But all our fusion attempts at Danish room temperature on a good summer day have been not very successful. The green bars is actually the fusion, and the gray bar is the leakage. And all we had seen was a little bit of leakage, and essentially all fusion was well below the threshold of 5%, which we said, where we said this is just background and we can't really characterize that. There's still a little bit more than some of the controls, but not enough that we would regard this as any sort of real-world content mixing. Things changed massively when we increased the temperature, and this is something where you actually need a chemist to do that, because we are the only ones who solve most of our problems with heating stuff. And when we moved with some of the systems from 20 degrees to 37 and 50 degrees, and our liposomes are perfectly stable, you can see there's a little bit of background on unmodified liposomes. There is essentially no background fusion as soon as you have linas on the surface. And this is easy to understand because each of these lina molecules carries quite a number of charges because the DNA has a phosphate backbone. And these charges ensure that the surface is slightly negatively charged and they repel each other, and that suppresses any sort of background fusion. But only in the case of complementary linas, and not all of them, but only a very selected subset, we saw for the first time, to our utmost delight, some substantial increase in the fluorescence signal, which ultimately is a representative of content mixing. And at the time, around 2016-17, that was an unprecedented level of content mixing. And have a look at the leakage as well. There's essentially no leakage. It's well below 5%, even by temperatures all the way up to 50%, which was a lot more than we expected. So the hot fusion results were very, very encouraging. And then we thought, is that something which is intrinsic to DNA, or could we actually achieve something similar with other oligonucleotides and different peptides, backbone structures? And we did some experiments with PNA-based structures. And since we do a lot of automated synthesis in the group, that was something which was within our reach. And we made some zipper designs based on PNA, and they were actually doing very well at 50 degrees as well. At that time, there was nothing near 30% content mixing. However, content mixing is not nearly as good as for the best DNA systems, which I don't think is based on the PNA and is just a result of that we have done a lot more optimization for designs and membrane anchors on the DNA side than we have done for the PNA side. But it does work, and then we have looked also at, you could call them hybrid designs, and this is actually peptide nucleic acids which use a DNA or RNA bridge. And again, you can see for the RNA bridge, you approach very, very substantial level of content mixing, and again, leakage is well controlled below 5%. But again, you can see there are substantial differences if you go from an RNA bridge to a DNA bridge, because the duplex type changes, and in this way, also the sipping action and obviously the energies involved in the hybridization change, and that has an impact on the outcome. So far, all the experiments you have seen have been done with typical liposomes on the small end of the nanoscale, 50, 100, or a little bit above, very tiny nanoparticles. And we sort of thought about, is there any scaling factor? Is that something which can be applied to particles which are much larger? And then we went to GUVs, giant unilamellar vesicles, around 25 micrometers. And as you can see, this is a factor of 100 at least in terms of size. And we used essentially a two-dye system. So we had one population with a red dye and another population with a green dye. And in case of content mixing, that would likely result in a typical mixed color, which should be easily visible under sort of a confocal microscope, since these particles are so big. And again, to our delight, that was exactly what happened. Even with a moderate amount of experimentation, we ended up having 50% of the limited population fused. And you should keep in mind here that GUVs are not particularly fast in traveling in solution. They are very big, they have a very low speed of diffusion, and that's why you don't see quite as much content mixing as you would see for much smaller particles. And just to convince you, there was no fusion whatsoever for non-complementary oligonucleotides of the same structure with the same membrane anchors. So you absolutely need a fully complementary sequence. But all you have seen so far was fusion with a single step and two populations of liposome. So our immediate next question was, can we go any further and go to 2, 3, and 4 steps? Again, this is something which comes from a chemist's mind, because we always think about multi-step reactions. And when we look at liposomes, we see compartments for chemical or enzymatic reactions. And that's what we tried. And we made sort of pairs of linas, which are only self-complementary. So there's always A and A prime, and B and B prime, and C and C prime, and so on. So fusion can only occur in the programmed order. And this was, as indicated in the title, that was the ultimate goal for this project for us. And that, of course, limits any sort of activities, to the best of my knowledge, to oligonucleotides, where all this programmability by hybridization is already in the molecule. So, and we have followed this process, and we have again used the SRB assay. And if you think about it, dilution is the dilution factor is highest when you start with the dilution, and gets increasingly less for any subsequent fusion event. The dilution factor decreases. And this is also what you see. But if you sort of calculate that to some sort of fusion yield, you can see that the fusion or the content mixing yield is actually quite consistent over the 3 steps. And for this DNA sequence shortcut TXP3, that means we consistently between 60 and 80% content mixing, which was very impressive at the time. So this is sort of half-time, I would say. So what we had in our hands was a platform for controlled and programmable biomembrane fusion with synthetic membranes, that is. We could efficiently control fusion at 37 and 50 degrees. We had nanomolar concentrations which were effective. And from some single particle studies, we actually deducted that we need around 10 or less double-stranded LINAs per particle to actually effectuate a fusion. Low leakage, fully programmable, and no obvious size restrictions going from a length scale anywhere from 50 to 25,000 nanometers. So what was next? We were very happy that things worked the way they worked, but we were still not quite sure if there's a subpopulation which does all the fusion. Do we have single, double, triple fusion when we do this? How many of the particles participate? Is there any curvature effect? And all these questions which would typically come from someone with a biophysical background. We teamed up with a group which we had a basic research center together, a physics group of Nikos Sasakis. He's an expert in both biomembranes and microscopy. And he collaborated with us on turf microscopy. And we immobilized tens of thousands of individual particles on a surface and followed them in different channels, whether we could observe fusion events or not. I have to make a little disclaimer. This is a very shortcut version of the whole story. You can see the reference down here. A lot of techniques and tricks had to be developed from the Hasakis group to actually do the barcoding of so many particles. Obviously, with the usual couple of colors, this is impossible to follow. So they invented a type of RGB encoding, very much like you would do for your monitor, where you combine ratios of dyes in a 3-dye ratio code. And this allowed us to barcode tens of thousands liposomes at the same time and follow them. So, and if you imagine how many combinations you have, this is really staggering as soon as you go to a certain sort of number of fusion events, how many variations you have. You can see the green, the red, and the blue channel, and this is very interesting. This data may not look like much, but it's actually very good. And thanks to Mette, which had been a PhD student in Nikos group at the time, she developed the machine learning algorithm to see the difference between an actual fusion event and what biophysicists call a kiss and run event. These spikes, which you see in the different channels, these are liposomes which approach each other and have maybe even contact, but they diffuse elsewhere very, very quickly. And only an irreversible fusion leads to a step and an increase in the particular channel. And this program can distinguish between the two and clearly see what fuses in which order, how many times. And that was really impressive to see that we could barcode the whole thing. And again, Meta has developed the software to do so. And you can see the different clouds which represent individual dye combinations which can be distinguished from each other, and many more than you would have available with just using dyes available. So sorry, that was a little bit too fast. The interesting outcome was that we could establish a model, so the data from these tens of thousands of liposomes could be analyzed in parallel. And what was the outcome? The outcome was that we see single fusion as the major event. That was expected. But what was not expected was that two fusion events are almost as likely as one. So we see 5,000 events. Even a 3-step fusion is by no means unusual. It's still in the same range. And it falls off with 4 and 5. But still, a lot of events are recorded. So this was actually very interesting to see. What we also established was that There was no leakage in the microscopy experiment, so it was essentially quantitative content mixing. At this point, already at 37 degrees, was sufficient, no leakage, and there was a very little background for sort of for non-complementary DNA, almost invisible. We also added our first enzymatic reaction with sort of fluorescein, which is glycosylated and only becomes fluorescent by deglycosylation by glycosylase. And but this way we could follow having the glucose, the FD-glu in one of the population, and the enzyme which actually cleaves off these sugars and releases the free fluorescein was in the other population. And in this way, we could clearly show whether things fuse and whether chemistry actually occurs. And all data is based on this. So the question was, based on this very exciting result, was how far can we go? And for those of you who don't have the numbers in their head, 100 nanometer liposome is around 400 septoliters. And what is a septoliter? You can do the math down here. All of you have heard about micro and nanofluidics, but being able to fuse and control populations of individual liposomes is still many orders of magnitude lower in terms of how much liquid you actually handle during these events. So we also wanted to apply that to some sort of real-world chemistry. And we decided for carbohydrate mimetics. There are a lot of applications of carbohydrates. I've just shown you from a literature. Some they are in all fields of drugs. You will find some carbohydrate-based candidates. And the problem is, for peptides, you have automated synthesis. For DNA-based and RNA-based stuff, you had automated synthesis. But for sugars, there's actually no automated synthesis. And so there's a big interest in having an option to do controlled libraries where you can actually program what is supposed to react with what in which order. And that's sort of the background for this very busy slide, and you're not supposed to look closer. This is just a classical combinatorial slide. You start with a starting sugar, then you have small libraries of B1, B2, B3, and then you just go along. So what you would need would be sort of a library system where you could do maybe up to 5 steps. Why only 5 steps? Because typically for small molecule drugs, you have a cutoff of 500 Dalton. So even after 2 or 3 steps, you will hit the 500 molecular weight barrier. So you actually don't need that many chemical steps. If you go into adjuvants and oligosaccharides, that is of course changing the field. But it would be very interesting to be able to make carbohydrate mimics. But before we can do any of that, we have to show that we can do 1, 2, 3 steps in liposomes and follow the progress of the chemistry. So we have picked mono, di, tri, tetra, and hexasaccharides and looked at their performance in the context of program chemistry. So we have combined this, of course, with a click reaction and associated dye, and the dye is only added to be able to quantify the chemistry afterwards by HPLC. So ideally, we have a LINA triggered fusion, and obviously with non-complementary no fusion. In the case of fusion, reaction partners will meet and may eventually react. And after lysis, products are so diluted that they will not be able to do any chemistry. This is a very important aspect. If you lyse the liposomes, then you dilute your content to nanomolar concentrations, and there is just no chemistry going on at nanomolar concentrations or below, at least not on the time scale we are looking at. We have picked an interesting group of molecules, but we have actually overlooked that sugars are actually pretty good, especially mono and disaccharides, to permeate through bilayer membranes. So a lot of optimization, which I will not show, has been put into optimizing the membrane for chemistry in a way that you can still do effective fusion without losing material inside for small molecules, which diffuses through the bilayer. So therefore, we don't see any chemistry for the monosaccharides because they diffuse out much faster than they can do chemistry. But disaccharides and all the other members give very, very decent yields and clearly react. We have different heating modes, but these are all details which are not too relevant. Key from the slide is that as soon as molecules stay inside, they will react and you will be able to follow that. We have expanded this to multi-step reactions, and you add the dye to the first sugar, and you add another sugar to the glycosylated dye, and you can see all the products in the actual HPLC trace. So you see your starting sugar, you see the monoadduct, and you also see the product. Obviously, with an overall yield of 16%, this is not very impressive, but take into account that this has been some very early results, and actually the first examples of doing sugar chemistry multi-step in very small compartments overall. It says in submission, this is actually has been published about a year ago in JACS, and we have even received a friendly email from the editor-in-chief that he liked this sort of research. So that was classical chemistry. Obviously, we wanted to look at enzymatic chemistry as well, and have again taken advantage of our favorite FD glue molecule and the glycosidase, have encoded, and now wanted to see whether we can do multi-step reaction and load more substrate and subsequent fusion events. And again, for those who have forgotten about that, this is essentially fluorescently silent, a very low background. As soon as you cleave off one, you get a weak fluorescence, but in case you cleave off both of them, you get a nicely fully fluorescent fluorescein as the one you know from microscopy experiments. And then we go on and do first step. The gray bar down here is sort of the control, and the second one, and the third one. And you see the time scale is very slow. That has changed a lot for other unpublished results. So we are now down to 2 hours for the full 3-step cascade. And this control has also flattened out. I've just not managed to get all of these results within. But even this, as it is, looks pretty good. So 3-step reaction with an active enzyme is possible. All of that, what you have seen so far, has been done with synthetic lipid bilayers. So a smooth surface, and the only other thing on the lipid bilayer are actually our oligonucleotides. And since concentrations are very low, the surface is largely empty, and that's why they are very amenable to fusion. So there are no mechanical barriers. All of you know that cells and other natural particles, and you will see if you look at the headline, extracellular vesicles, which are like very small cells, have a lot more stuff on the surface. All the proteins, all the glycolipids, anything which you will typically find on a cell surface, you will find on exosomes as well. And extracellular vesicles have been very popular because it has been accepted now in literature that they have very important function and in signaling. And the most prominent example might be stroke exosomes, which communicate with the brain, can cross blood-brain barrier, and prevent your brain from damage by a loss of oxygen or lack of oxygen. And we have teamed up with the molecular biology group to look whether our concept can be extended to natural lipid nanoparticles, such as extracellular vesicles. And in this particular case, exosomes, which are sort of a small the small-sized subpopulations of extracellular vesicles, which otherwise come in different sizes. The important part is there's an incredible number of these EVs as they are shortened in your bloodstream. Essentially, all cell types release them. They are certainly not what was in literature in the old days, sort of the trash can of the cells. All of them have functionality. There are sort of good ones and bad ones. And especially two more exosomes or EVs are very interesting because they are suspected to be involved in preparing other places in the body for metastasizing. And it has also been shown that exosomes contain genetic material, they contain microRNAs, and it's very interesting to see what else. And even on the surface, RNA has been spotted. Okay, so in contrast to this nicely clean synthetic liposome, exosomes have a lot more stuff on the outside, and we wanted to know whether they are still amenable to fusion. I will not go into details of the biology. This is not very relevant. What we wanted to do was is depicted here, and we wanted, of course, to show that we actually do content mixing. And as soon as you work with cells, this is a little bit more difficult. So we picked the most difficult of all contents, sort of an mRNA, which is a little shy of 1,000 mer of RNA. So it's a relatively unstable molecule. You know that from your COVID injections. And fuse it with some extracellular vesicles, which have been isolated from mice. And ultimately, we would like to have this hybrid particle, sort of a biomimetic EV, and then deliver this one to cells because it's known that they are taken up by other cells. And ideally, our mRNA would be expressed, in this case, not to a spike protein, but it codes for mCherry protein, which is very fluorescent. And in this way, we can follow whether the cells actually take this up. There are a lot of technicalities in isolating EVs, purifying that. They have been sort of immobilized on magnetic beads, also by hybridization. There has been sort of a DNA-controlled release of the clean hybrid particles. So you do the fusion, you wash off all particles which have not fused, and then you release in an additional step by hybridization sort of these EV hybrid particles, which are supposed to contain the mRNA. This has also been published in JACS this year, and we are very happy about the outcome. They have been sort of upscaled on magnetic beads. The problem is typically you have only 10 to the 9, 10 to the 10 particles. This is very low, and these magnetic particles allow it to upscale that with a factor of 1,000,000. But what are the results in terms of cargo delivery? And as you can see, I will guide you through these busy slides. So the target were some HEK cells. And as you can see, we have a nice mCherry signal for complementary LINA fusion. So that actually works. Liposomes alone, non-complementary, and EV controls show no signature whatsoever in the red channel for the mCherry. And the only thing which gives a very, very strong signal, even more than sort of our fusion signal, is a positive control from a delivery agent. And still, we are super happy with the result because these delivery agents, like lipofectamine and RNA max and similar, these are polycationic amines. They are very toxic, and they can only be used for cellular systems. So you can't use them in animals, and you obviously can't use them in patients. So you can deliver something to a cell, but your project is at a dead end at that point. Whereas our LINA fusion, at least theoretically, could be upscaled and could be used in animals as well. And this is also something we are very interested in. So that was the end of the story for biomembrane fusion, going from no fusion when we started to programmed multi-step fusion. and ultimately to fusion of with natural particles, and we have been very happy about. So this is a little bit of a vision slide. So we really work now and have some PhD students working on chemical and enzymatic libraries, and we try to do multi-step reactions, 5, 6, with fully encoded particles. Remember, each lina on the surface is like a barcode, so you can read out at the end what has reacted with what in which order. Then we have still a very pronounced interest in transferring some of these techniques to a microscopic setup. In the turf setup we had, we could look at 40,000 individual nanoreactors at the same time. So this is really something which is amenable to upscaling. We are interested in exosome diagnostics because we are very interested to look at delivery of things from these hybrid particles, because this is the mildest of all available methods where you don't invert anything on the surface to the inside, you don't leak anything out, and there's no other technique available to actually modify the content of such particles. And of course, we always look for new ideas and collaboration. If any one of you has good ideas, you're more than welcome. Obviously, I've been very much involved in the early days, and I'm still very much involved, but a lot of the practical work and a lot of the ideas are coming from people in my group. And this is a slide of my early fusion team. So these are the guys which have actually enabled fusion going from 0 to a really good content mixing, especially Oliver Rees, which has been one of my first postdocs on this project. And Alexander Rabe have been very instrumental. He has worked with the PNA. They both work for BioNTech, which was one of the big players in the so-called BioNTech Pfizer vaccine. And all the others have found nice jobs in biotech. This is the current team from last year from one of our groups, Dinners, and Nasmir, she's working on the exosome project. Luisa and Nikolai are chemists working very hard, and Lu Fang has joined our exosome and enzyme team. There are, of course, external collaborator with the CHEMS group on the exosome project, Nikos with the microscopy, and the Xodo group for some of the therapeutic applications, and obviously my own group as well. There are some foundations which need a big hand. Velux Foundation, which funded some of the initial work, the Novo Nordisk Foundation, which I'm very proud to be a Novo Nordisk Not Fellow for the upcoming 5 years, and the Lundbeck Foundation as well. And now the interesting part, you are most welcome to ask any questions you may have. And this gives you an impression how a cloudy winter day looks in Denmark. And thank you for your patience so far.
Thank you so much. That cloudy day is really sad. No wonder you're staying in the lab. There's quite a bit of questions in chat. Can you see chat?
Yeah, I will go to the chat.
If you could read a question out loud,
Yeah,
that's going to go for the recording.
Yeah. So just a moment. So the Happy New Year is obviously not a question. And let's, yeah. So the first question is, how did you optimize the amount of DNA anchors per lipid? So the DNA to lipid ratio, or does it work on a wide range of ratios? We have optimized that very empirically, I have to admit. So we have just covered all imaginable ranges, have settled with sort of a range which both ensures a fusion, and because this is we do micrograms of these lipidated DNAs. So this is not something which you have in a bottle. You have sort of a limited amount. And that's why we have to try to find a balance. You can get a little bit more fusion when you go to a higher ratio, but it's actually flattening out very rapidly. So there's no big benefit in having more DNA on the surface. Because obviously, if 5 to 10 DNAs can't do the trick, 100 won't do either. So it's at a very low concentration. So, and the next question is, how do you actually calculate the efficiency of the fusion, and what percentage of the population fuses? I have to split my answer here. For the bulk experiments, we are not able to talk about a particular population because we can't see whether some have fused multiple times or everyone just once. But for the microscopy, we know that we have that the whole population fuses. We have seen from the 40,000 immobilized, 95% of all of them engaged in at least one fusion event. So it does not seem that there are subpopulations or curvature-related issues which play any particular role here. They all do fuse. And the actual calculation of the efficiency for the fusion, which I translate into how do we actually calculate the content mixing, this is done with the SRB assay for all the classical measurements, where we have sort of 100% when we do a full lysis of the SRB population. This will be the maximum fluorescence value you can get out. And then we put this relative to the fluorescence signal we get without the lysis. And we do exactly the same for the leakage experiment. So the shape of the liposome after diffusion is actually very interesting. We assume, and I particularly use the word assume, that these liposomes fully flatten out and just become one with the receiving membrane. And then, of course, for all our synthetic experiments, both lipid compositions are the same, so they are indistinguishable. But for the exosomes, we are actually in the process of looking at that by various means to actually see how fast is lipid mixing, do we have remaining patches like rafts on the surface or not? This is ongoing research, and hopefully next year or during this year, I will be able to tell you more about it. So I think the shape just remains spherical. We use multiple NTA techniques to follow the size and the surface charge of these liposomes, and also for individual liposomes. And typically, if they would adopt very unusual shapes, we would see blinking, and we don't see that. So as long as they stay roughly spherical, the signal will be stable. If they start becoming banana-shaped, the signal should fluctuate on an individual level, and we don't see that. And yeah, and of course, I'm very happy about those who have no questions, but have just enjoyed, yeah, the roughly 45 minutes of me talking about this very exciting subject. I still am thrilled what you can do with all of that. And there's one more comment. How specific lipid compositions? That is a very important question. Obviously, you cannot just fuse plain POPC lipids. You need a fusion mixture which has a certain fusogenicity. This doesn't mean that that you have a mixture which is super fusogenic and just fuses in the background, but you need a composition which allows a fusion, and you will typically have some cholesterol in it and some other component. But since there are so many lipids and so many combinations, we have by no means the ability to really investigate that. But we have a project on that, and we have also optimized our liposomes a lot in this regard to have the best ratio between fusogenicity and keeping things in the inside, which is linked to our specific interest of doing enzymatic or chemical reactions in the inside. I have, this is the last question I can see in the chat. We have, of course, for our first angewandte paper, we have compared the sequences from Höck and also from Boxer internally and have run them as controls together with other controls. And in our hands, none of them have shown any content mixing. They do lipid mixing, but we have not seen any content mixing for those. And the problem is, on a more technical level, companies are not even able to make them. We have received from, without calling anyone, but from a very big player in the field where we usually order oligonucleotides of other nature without any problem, and they have not been able to do them. The problem with those is that these companies, they probably oligos of this composition are in very low demand. So even though they are experts for oligonucleotides, they do very, very rarely synthetic oligonucleotides with lipid incorporations. And that leads that most of the lipids are in very, very low yield, much lower than you have ordered or they give you on a sheet. And most of the oligos of commercial nature we have received contained less than 10% of the oligo desired, and some of them were plain wrong. And the companies have all been willing to try again. So customer support was very good, but at the end, they were not able to make them. And as you may have seen, most of the cholesterol and tocopherol variants, they are all terminal modifiers, which is the worst setup you can have because they all become amphiphiles. So they are foaming so much. If you would have the money to buy enough, you could use that actually as shampoo. So, and we have deliberately developed building blocks which can be inbuilt technically anywhere in the sequence, and we have at maximum we are able to inbuild 4 of these building blocks, which means up to 8 lipids. But what is really difficult here is purification of these oligonucleotides. So there's a lot of knowledge is in the purification and to have a yield which you can actually use for anything. So right now, I don't think there's a company which can actually deliver these oligonucleotides, despite having some of these hydrophobic building blocks in their catalog. I hope this has answered your question. But I'm happy to take more questions, either from the chat or from elsewhere. And you are also welcome to drop me an email at a later point if you are not entirely sure how to phrase it right now. And I hope you find the subject interesting and can see your potential also in completely different areas like origin of life and artificial cells and stuff like that.
Thank you. Thank you so much. Thanks for going through all the questions too. I want to echo what you said. We're having the same problem getting some of those. We do some of the fusion experiments too, and it's a pain in the lower back to get any usable yields out of those syntheses. And yeah, we've been struggling too. It's annoying. We kind of switched away from the nucleic acid systems now, partially because I was fed up with getting it.
Understandably so. And this is not a lack of scientific skills. This is just very amphiphilic stuff. And the modifications available are just not very amenable to what people interested in fusion want to do with them. They are really designed for delivery purposes and not for anything else. But I will be happy maybe on a private chat at a later point to have a meeting if you are interested to sort of compare some oligonucleotides, which we would be happy to deliver if we know a little bit more about the system and to help out here.
I would love to try because we've been mostly using the cholesterol label to anchor an oligo in the membrane, and sort of works, but it's not ideal, and it's.
And I haven't talked much about that, but especially also the chemistry around the actual membrane anchor is very, very sensitive. And we have seen membrane anchors which look very nice on paper and they didn't do a thing. And it was all about sort of the next 5 atoms to the left and to the right of the membrane anchors, which make a lot of difference, obviously. The problem is, despite having done 100+ different oligo designs, this is still not enough to have any sort of predictive power. We have a good gut feeling, as it's called, but it's obviously this is not very helpful if you just want to use that as a tool and not sort of base your entire research on it.
Yeah, exactly. I think that's might be part of the reason why we don't see a wider adoption of it.
I think it's really, really difficult. And also a part is I have given a few talks to some biophysics meeting, people are very reluctant to approach us or others to actually do some oligos and try to do it themselves. And in this regard, some of the earlier publications are not helpful because they have sort of indicated you can do that with those and those building blocks, but in our hands, and I guess in most hands, they simply don't provide content mixing. And I assume that is what most people really want.
Mhm. Yeah.
Yeah.
Exactly. All right,
Yeah,
I'll shoot you
yeah.
an email and maybe we can
Yeah.
brainstorm further.
You're more than welcome to schedule a Zoom meeting for just the two of us or some collaborators from your group, and to see if there's any useful input from our side. We are happy to help, and yeah.
Awesome. Thank you so much, and thanks everyone else. It was a great
Thanks
way
for
to
your
start a
patience
year.
and for your invitation. And for me, this is end of the day. I think this is not quite the same for you. And I will bike out into total darkness and yeah.
Brave the
So enjoy the
wilderness.
rest of the day, and yeah, good luck.
Thank
And a happy
you.
New Year to all of you. Bye bye.
Happy New Year. Bye.